Right. Welcome, BioBuilder Club. I see. Thank you. Many of us were able to join us live here. And if not, hopefully you are watching this recording later. So we have the wonderful Danielle here, very own BioBuilder. He's going to go through biodesign this webinar. And I will say that we're going to be splitting up the biodesign process. And so we're going to cover enough to get teams started. And then later in this season, Danielle will return to give a second biodesign webinar to cover more of the parts level. So that way, once teams kind of have a project that they're working on, hopefully that'll be slightly more timely information in terms of project development. So do not worry, we will cover all of the levels of the abstraction hierarchy, and you can always email me at any time. We can answer any questions. So Danielle, I hand it over to you.
Hello, I'm so glad you all could join us, and those who are watching the recording, that is great as well. As Chloe mentioned, we're going to be walking through the first levels of the abstraction hierarchy, and then as you all get deeper into your projects, we will get into the finer nitty-gritty details of parts level design and DNA. All right, so, I'm trying to go through my slides and it is not moving. Are they moving on your screen, Chloe?
No,
No.
they
Oh, wait,
oh
here we
now
go.
they are. There we go.
Okay, I have to click. Sorry, I can't use the arrows. All right, so first we're going to define what synthetic biology is, or bioengineering. They're pretty synonymous. So synthetic biology is essentially a combination of two big areas of science, one being molecular biology. So we're going to be using molecular biology techniques to edit organisms' DNA, to give it some traits or modify traits, but we're also going to be looking at it from an engineer's perspective. So engineers like to design new things, new parts, new pieces, new systems, and then they test them, or sorry, they build them, and then they test them. So we're going to be combining these features of molecular biologists with an engineer. You can kind of think of it in terms of an interior designer versus an architectural engineer, right? So the molecular biologist is your interior designer. They're going to go in and just kind of like change the aesthetic or change the vibes of your place. Whereas if you want to completely renovate your building and change up the flow and the function of it, you're going to bring on the engineer, right? So we're kind of merging those two into one person and tackling scientific problems that way. Okay, so we mentioned we're going to be going through designing by abstraction. You can find a lot of the information we're going to talk about today and even more details in both your BioBuilder Club handbook that Chloe has written for you. It's an amazing resource. And in addition, your copy of the BioBuilder book. The first few chapters are also going to walk you through a lot of what is going to be covered today. All right, so this is basically the abstraction hierarchy here. We're going to go through topic area, system, device, parts, and DNA. These are the steps that you're going to work through in designing the project that you will end up presenting at the Big BioBuilder Club meeting in March. You can go through it from the bottom up, but typically at this level, at the high school level, you don't have quite enough knowledge of what DNA parts are already out there. So we advise more of a top-down approach. That's how we're going to be walking you through it today. So the top-down approach, we're going to start with what's the topic area that you would like to research and propose a system for. Okay, and you've probably already done this process before if you've ever planned a trip or a vacation for yourself or a group of friends or your family. You've already worked through these steps before. So for example, if you really like getting out there in the outdoors and you want to go backpacking somewhere, great, you've determined your overall topic, right? I want to go on a backpacking trip. Now you have to decide, well, where do I want to go? Do I want to go somewhere local? Do I want to go to the other side of the country? Do I want to go somewhere I've never been before? What kind of time do I have? Do I have the money to be going to another country? Do I have a passport? Once you decide those things, you've got your location in mind. What are you going to do there? What specific things can you do? Do you want to really challenge yourself? Do you want to have like a nice easy backpacking stroll. How are you going to do it? So once you get there, are you going to have to rent a car? Can you take public transportation where you need to go? How are you going to time your meals? Are you going to stop and camp along the way, or do you need to set it up so that you're getting to hotel points? And then finally, what are the specific items that you're going to need for this entire trip? Okay. So this is kind of the level of thinking that you would go through when planning a trip. And that lines up perfectly with our abstraction hierarchy, right? So as you work your way down, you're getting into the finer and finer details of what it is you need to achieve your goal. And this is what you're going to be doing for your projects. So for today, we're just going to be focusing on topic area, system, and device level of biodesign. So we're going to walk you through how to identify the topic area and the problem that you would like to actually address. We're going to talk about how to brainstorm approaches to this challenge and create a system or propose a system that would allow you to address your problem. And then we're going to talk about device level design. So decide on the actual approach of how you're going to do it. In our second webinar, we will get into the finer details of parts. We find that a lot of students are pretty new to that part. So we like to spend a little bit more time working with you all on that. Okay, so topic area. There are so many different topics that you could address with synthetic biology. I'm really bad at deciding on topics. I get decision paralysis. There's just too much to do, too many cool things. A great place to look if you're stuck, I put right here is the iGEM website. If you go to the iGEM website and go to their villages, they give you all the different broad topics that your project could cover. And if you click on each of those, it'll even give you finer ideas that fall within those. So these are all amazing choices. I will say we do tend to get a lot of health and medicine projects. Obviously, there's a lot to tackle there still, diagnostics, therapeutics. We also tend to see a lot of environmental things. Again, there's so much to do there as well with biosensors and such things. Lots of projects covering plastic. Plastic is a huge problem. But don't forget about some of these other cool things, like the food industry is huge, nutrition, lots of stuff there. Cosmetics is a really cool area where synthetic biology is really taking hold. Ginkgo Bioworks has done some work with fragrances, materials, using spider silk for various materials such as bandages. Agriculture is a huge area that's starting to use a lot of synthetic biology. So the possibilities are really endless. But I do highly suggest going to the iGEM website just to kind of get some ideas of what's out there. Okay. All right, so that's your topic area. So we're going to work through an example as we go through these different levels today. So let's say that you want to design a cell that can be used as an environmental biosensor. Let's do that. Okay. So that's our overall topic. Let's get a little specific. So let's think about what we might want to detect out there. Arsenic contamination in drinking water is a huge problem. You tend to find it's naturally in the soil, but it's a really big problem in areas where there's a lot of mining and fracking, where it can release that arsenic, and it can concentrate in the groundwater. It's a heavy metal, so it is very toxic to humans. It is an issue in the United States. There are pockets of it out here in like Arizona and California, and then over in the Northeast, we also see a lot of arsenic. It's also an issue worldwide. You can see the areas with the orange and red throughout the world, really, it's everywhere, have high levels of arsenic contamination in their drinking water. So this is an issue that would be important both locally, as well as the whole country, as well as worldwide. Okay. Okay, so now we're gonna get to system area. System area is gonna take you a while. This will take you some time to work through because there are really three things that you need to consider when thinking about how you're gonna build your system, okay? And we're gonna work through all three. So first is, how do you want your system to behave to address the issue that you have selected. The second is what's going to be your input for your system. So what's the thing that's going to trigger your system to do what it's going to do? And what's going to be your output of your system? What is your system going to do? And then finally, what cell type? We just have a miscellaneous bacterium here. So what will be the cell type that will work best for what you are proposing to be done? Okay, so we're going to work through all three of those right now. All right, so system level design, problem number one, how do you want your system to behave to address the problem you have selected? Okay, so this is our kind of diagram of our overall system, right? You might have a diagram that looks like this on your poster, for example. So you'll have your cell, you'll have your input, and you'll have your output, right? So we're gonna start real basic. What is it that you want your system to do to address your problem, right? So you're basically gonna come up with a statement where our group wants to engineer a cell that blank, and then fill in what your cell is going to do, what your system's going to do. You don't have to be very specific at this point, okay? Let's go through some examples. So if the problem that you propose to address is pollution caused by plastic containers, if you've gone on a walk or run outside at any point in time, you've probably encountered plastic trash on the ground, right? Or in waterways. So if this is the problem you want to address, your system, your living system could just be engineered bacteria that can break down plastic, okay? We're just at a very basic level at this point, okay? Another example, let's say the problem you want to tackle is gluten intolerance. The living system that you propose could be an engineered bacteria that breaks down gluten in your body, right? All right, let's go with our chosen one for our example. Our problem that we want to tackle is tap water contaminated with arsenic. Okay, we want there to be some way for people to know if their tap water is contaminated with arsenic. We don't want them to drink it. So the living system that we are going to propose could be engineered bacteria that produce some sort of a visible signal when arsenic contamination is detected in the water. Okay, so see how these are different? Our endpoint is kind of different. So two of these, the endpoint is to break down something. And our last one is to produce a visible signal, like a color, right? Okay. So that's your first part of system level design. What's your system going to do? Now we want to get into a little bit more finer detail. We're going to look at this input and output part. We want to know what the signal or input is going to be that will trigger this system to do what it is we want it to do. And then we're also going to define what it is we want it to do, and that's going to be our output. And an important thing to note here that students get a little tripped up on is the output should be something that's measurable. Okay, and we'll give some examples of what we mean by that. So now we're going to take our generic response of engineer a cell that blank. And now we want you to be able to string together a more detailed sentence. For example, engineer a cell that does Y, which is your output, in response to X, whatever your signal or input is. Okay, so let's go through some examples. And I would love for this to be interactive. I know it's hard. It's Monday evening, but I've got the chat open, so feel free to use the chat. So let's practice defining some inputs and outputs. Let's say our problem is that first one, pollution caused by plastic containers. Our solution was engineered bacteria that break down plastic. So what might our input be? Well, our input here could be plastic, okay? And as you get finer into your research, you might determine specific molecules within the plastic. That could be your input. But here we're just talking real basic. So our input would be plastic. Plastic would trigger our cell to then produce something that breaks down or degrades the plastic. Okay, so our input would be plastic, our output would be our broken down and degraded plastic. All right, let's do another example. Let's see if we can get some people going in the chat here. Let's say our problem was the gluten intolerance. We want to tackle gluten intolerance. Our living system is engineered bacteria that can break down gluten. So what might be our input here? What's going to be our signal that triggers our engineered bacteria to break down gluten? All right, we got one in the chat. Thank you. Yes, good. Our input is just going to be gluten, right? Our output, what might our output be? What do we think our output might be? Input is gluten, so gluten is going to trigger our bacteria to do what? What's going to be our final output there? Great, good. Yeah, so a product of broken down gluten. Yeah, definitely. Or a signal that shows gluten sensitivity. Yeah, good. Yeah, I see some votes for that one as well. Good. Okay, our last one. Let's do the example that we're working through. So tap water contaminated with arsenic. We want to engineer a bacteria that's going to produce some sort of a visible signal when arsenic contamination is detected in water. So what would our input be here? What's going to trigger our bacteria to produce that signal? Good, yeah, arsenic, yeah. So arsenic is going to be our input. What's going to be our output here? What's our output? We're going to test a water sample. If arsenic is in there, it's going to trigger our bacteria system to do what? Yeah, it's going to signal. So we've got a visible signal. What's an example of a visible signal that we might be able to use? Color, good, love it. Yeah, yeah, so it could give off a color. Great. Now, at this stage, you don't need to be specific about the color. So please don't spend entire BioBuilderClub meetings arguing over what color your system's going to generate. If it's going to be a color generating system, you can just be generic with it right now and just say color. Okay, our output is going to be color. Because you might choose a color and then later on think, oh, that actually won't work in the environment that our organism needs to be in, or it wouldn't be super visible. Okay, so just color. Okay, so we're on our last part of system, and this is where system level design gets a little tricky. You're going to need to decide what the best host cell, or we call it a chassis. You pronounce this word chassis. So what's the best chassis or host cell for the genetic program that you need to use? This is going to be a really important step in your project, and you might change it several times. Okay, this is where you're really going to have to start doing some research. But I'm going to show you some ways to kind of narrow down your choices to what chassis might work best for you. Okay. So you want a chassis, this seems pretty obvious, but you want a chassis that's useful. It has useful qualities for whatever your project is or what your proposed system is. If it's something where you need it to grow a lot, can it be grown really easily in a lab, or does it have really finicky requirements for growth? Some microbes are really tricky with the temperature that they like and the type of nutrients that they like, but some are really easy and they'll grow pretty much anywhere in whatever conditions. You also want to pick a chassis that has a known sequence. So we can look up its genetic sequence. That way, when you get to the parts and DNA level designs, you will be able to more easily do that. It's going to be really hard to genetically engineer an organism where we don't have its genome sequenced. Okay, so you want something where the sequence is known and it's easy to manipulate. It's been used in labs before. This one seems pretty obvious, but you want to make sure that it's not harmful to humans or other animals. Some of you might come up with a project where you're going to be releasing your system out into the ocean or a pond or soil, and you want to make sure that it's not going to harm anything, right? A tip I like to give students is to think about the conditions of the environment where your cell is going to need to function. So where is it going to be? Oh, I got a question. Where can you find the genome? We will get more into that in the second webinar, but you should be able to easily look that information up. And we can send some more information out about that as well. But that's a good question. Yeah, so you want to think about how are we going to use our system, where, and what conditions will our host cell need to survive in. Okay. So the easiest way to pick a chassis is called the nearest neighbor. And that just means you're choosing a chassis that already has an ability similar to what you have in mind for your system. Okay. So for example, let's say you want to engineer a cell to convert plastic into electricity. That'd be pretty awesome, right? Solving two problems, right? Getting rid of plastic and also producing electricity for us, which is another issue. Well, the good news is there's already a bacterium. Let's see if I can get this name right. I had to look it up. I have to move the chat so I can read it. It's covering mine. So there's a bacterium called Shibonella onidonesis, which is an aquatic bacterium, which already has the capability to generate electricity. It also has a few other really cool qualities, but it already can generate electricity. So this might be a good bacteria to use if you want your system to generate electricity, because it can already do that. You just need to add in the detecting part for your input, right? Pretty cool. And this bacteria is aquatic. So this is where you might say, okay, maybe we're going to focus on plastic that's in waterways because this microbe is aquatic. If you wanted to tackle plastic in landfills, for example, and produce electricity there, you might not want to use this as your chassis, right? Because it's an aquatic bacterium. You would want more of like a soil bacterium for that. Okay, let's do an example. Let's get that chat going again. So let's say you want to engineer a cell that will help degrade plastic debris in the ocean. And the biggest plastic problems in the ocean tend to be in the upper levels, kind of near the surface. Let's keep that in mind. So what features or characteristics would you look for in a chassis to engineer for this type of a living system? So list some traits in the chat. What are we thinking? What might our chassis have to be able to do if it's going to function in the ocean to degrade plastic? What do we think? Something that could break down plastic, possibly. We might also have to engineer it to do that. But if you want to release some sort of cell in the ocean and you don't want it to die right away, because that is not going to do much good then, right? You want it to be able to survive in the ocean. What types of characteristics does it need to have? What does it need to be able to do to survive? Make sure the product has an aquatic background. Okay, so whatever it forms, because obviously it's not just going to make the plastic disappear, right? So the plastic has to be converted into something that's not harmful, right?
So this is more about, oh yeah, there we go.
There, okay, we're getting closer. We're getting closer. Yeah. So it can survive in cold water. Yes. Excellent. All right. Hopefully that gave people an idea. We've got a lot more. What other qualities would it need to have to survive in the ocean? So it can survive in cold water. Great. What else does it need to be able to tolerate? Maybe that's a better word.
Yeah, so like the cold environment, it's more about the environmental conditions than what you're actually trying to get it to do in the end.
Yep, amount of sunlight, that would be something to consider. Yep, so do we want it to be photosynthetic since it's close to the surface? Good. pH, excellent. Yep, we got to consider pH. Salt, fabulous. I was just going to give a hint about that one. Yep, so it's got to be salt tolerant for sure. A lot of bacteria are not, and it would just die immediately. Let's see. We're releasing a living cell into the ocean. So what's something that we want to be really careful about in our selection process? Once it's out there, it's a little hard to kind of release it. Okay, so can it kind of like self-sustain out in the ocean? Yeah. What might you worry about when you release a living thing out into an environment? Oh my gosh, the motion lights just went off. Sorry. I got to move around a little here. That's a fun one. Okay. What else? What else? What else? Yes, good. Make sure it wouldn't harm other. You guys are getting a BioBuilder tour here as I turn on the motion lights. Yes, no one eats it. Okay, we got it. Yeah. Yeah, so we want to make sure that this is a microbe that is not harmful to any aquatic organisms at any level, right? We have the example here of the whale shark. They're kind of like filter feeders, right? So they are definitely going to eat some of our microbe at some point. All right, I think we got them all. So I'm going to throw mine up there. Yep, so we got the cold tolerant, salt tolerant, pH, photosynthetic because it's close to the surface. It should be one that can kind of like float and go with the current or swim. We got that it should not be harmful to marine organisms. We got the plastic digestion capability. We want it to be something that's well understood already. And the other thing that a lot of students don't think about is you can actually bioengineer your microorganism to have a self-kill switch so that it's only in the environment as a living thing for a certain period of time, which would be important in a situation like this. Okay. And we can get more into that in the parts webinar. Okay, so cells are annoying to deal with, right? They are living things. Living things are annoying to deal with. They are unpredictable. If anyone's ever worked with little children, they're very unpredictable, right? Bacteria and yeast, all those microbes can be the same, right? Even if it's a strain that we've used in the lab tons of times before, there's always going to be some variability. So another option, which is becoming more popular in synthetic biology, is something called a cell-free system. And these are pretty cool. So we know that in order to get a protein produced, we need the cell to go through transcription and translation, okay? And the cell does that by using all these molecules like RNA polymerase and ribosomes and tRNA. Well, we can take those cell guts, the cell lysate, and put it in a tube. We can kind of like mash up the cells and just take the inside bits, put it in a tube. We've also got energy sources in there, some ATP. And now, instead of having to worry about growing these cells in the lab and giving them the right nutrients and the right temperature, now we just have a freeze-dried pellet of the bits that we need. So you can just directly add your engineered DNA, and all of those cell machinery pieces are going to transcribe and translate it and produce the protein of interest. Produce whatever protein it is that you want it to make, okay? All right, so what are some benefits of doing this? Aside from not having to work with the living organisms, which can be annoying, it's a pretty high throughput process. And I want to be a little specific here. What I mean by high throughput here is that it's fast. You don't have to wait a few days for the cells to be streaked out on plates and then make a liquid culture and then grow a bigger liquid culture. It just happens within a few hours in the tube, okay? So it'll be pretty fast and that makes it low cost. The trade-off here though is it can't produce protein at super high levels. So if you have a project, like some sort of biomaterial that you want to produce, and you want to produce a lot of it, like synthetic silk, this is not the way you would want to do that. You would want to do it in actual cells so you can scale it up and produce more and more and more and more. Okay, but this is a fast and cheap way to get it done. Okay, another benefit is you don't have to worry about the production of any toxic proteins that would kill your living cell, right? Or maybe the protein that you want to produce is modified so that it's not natural and wouldn't be able to survive within the cell. Okay. They actually had that problem when they were developing synthetic insulin the first time. Obviously, they wanted to produce as much as they could, but it was making the cells literally explode. So they had to kind of like scale it down. Okay. The other really cool benefit is that because these are freeze-dried pellets, that makes them very easy to store. They're not really temperature dependent, right? And you can easily distribute and use them out in the field. So because of those qualities, these cell-free systems are really good for uses in environmental monitoring, like biosensors, food safety, medical diagnostics. So things where you could just test it within a matter of hours and you can take it with you out in the environment. This image is just showing you that cell-free systems, the sensor output could be lots of different things. So it could be some sort of fluorescence biomarker where you apply your sample to the cell-free system and your output is a fluorescent marker. It could also be colorimetric. It could be microfluidic. It could be electrical chemical. So there are lots and lots of possibilities with a cell-free system. So that's out there. Okay, so we've done our topic area. We've done our system design. Now we're going to get into device. Okay, so device is when you actually start thinking about how is this really going to work? So here's where you need to think about what do you need to make your system's behavior happen? Meaning, what do you need to detect your input? And what do you need to produce your output? And by you, I mean your cell. I mean the cell that you are engineering. Okay, so when we draw this out, we like to draw our system here, our input, our cell, our output, but inside the cell now we have this black mystery box, okay? And that kind of represents our device or the mystery magic that needs to happen within our cells. Okay, so let's do some examples. All right, let's go back to our examples here. So our example of engineering bacteria that break down plastic. Our input's plastic, our output's going to be degraded plastic. So here our device we could just call it a plastic degrader, right? We don't know what exactly those pieces are. We're just kind of defining what we want it to do, what that black box needs to do. Okay, let's do another example, then I'll ask you guys to do the last one. Let's do our gluten intolerance. So our input's gluten, and we want our output to be broken down gluten. So that mystery box inside of our cell, we can call a gluten degradator. Okay, it doesn't have to be super specific yet. All right, let's do our arsenic one. So we want to engineer bacteria that detect arsenic and then produce a color signal. So what might our device be? What do we need our cell to be able to do? Throw it in the chat. What do we think? An arsenic detector. Excellent. Yep. What else might we need? So it can detect arsenic. Awesome. What else does it need to do? So we take our sample of water. let's say it has arsenic in it. Our cell has an arsenic detector in it. Great, it senses that arsenic. Now what? Our output is a color signal. So what other device do we need in our cells? What could we call it? We got this, we got this. I'm watching the chat here.
And this
Okay,
can
yeah,
be a separate device.
yeah, so I'm gonna show you in a second. It could be, oh, we got another one. Okay, yeah, so you could include something like GFP, green fluorescent protein, so that when the detector senses that arsenic, it's going to glow. Yeah, so I think we got it here. Yeah, so we're gonna need a some sort of color, oh my gosh, sorry, everything's covering this. A contaminant detector like arsenic and a color generator, okay? And I'm going to show you in the next few slides how that could work, because there is no one way to do this. And as you're going through and designing your project, you might redesign it 100 times because there's no single right answer, okay?
And could
Okay.
you see, Danielle, just really quick, sorry, like just speak a little bit to why we're thinking of these at this stage as a black box and how that could be helpful? Just because sometimes that gets a little tricky for students.
Yeah, so again, we're just being really generic here and vague because we just need you to kind of wrap your head around what you need the system to do so that then you're able to go find the parts that you want. Sometimes if you just jump straight into parts, like if you did the arsenic one, for example, and you were like, okay, we're gonna detect arsenic, and then that's gonna trigger GFP to go off, then it gets a little trickier. If you just think of it more generally, like we need something that's going to sense arsenic, and then we need something that's going to give off a color, right? Then you can kind of get that basic design down, and then you can kind of finagle the details as you start to look it up. Did I miss anything, Chloe? Did you want to add anything? Okay, great. Okay, yeah, so again, we're being really basic here with the details. You can see it's not super professional to say like, oh, plastic degrader in our cells, in our mystery cells, right? So we're just thinking of it as a really general level, because you can get really bogged down in the details fast, and we don't want you to do that. Okay, so let's talk about the different ways that you can do devices. You could design something where there's one device. For example, with our arsenic one, you can just have one device, and that device can both detect arsenic and generate color. Or you could have multiple devices, and that's where it gets complicated really fast. That's why we're talking really generically here. So you can have two devices where one of them is able to detect arsenic, and that device signals the other one to then create color. Okay, I'm going to walk you through both of these options, all right? And there's no right or wrong way, okay? So in option number one, we've got just one device, and that sounds more simple, right? And just because it's one device does not mean it's the easiest way to go about it. This could also become complicated. So in this one device system, when arsenic is present, it directly turns on the color generating response. So in this system, color will not be produced unless arsenic is present, which is what we want. Great. You could do it this way. Option two, you could have two separate devices that give you the same outcome. All right. So here we have an arsenic detecting device. That device's job is twofold. It's going to produce some sort of signal that regulates whether or not the color generating device is turned on. Okay, so this arsenic detecting device is always going to be on, and it's going to be controlling the color generating device. So color will not be produced unless the arsenic input is detected by this arsenic detecting device. All right, now what in the world do I mean by that? I know a lot of that just went over everybody's heads. So we're gonna walk through what that looks like. If you have more than one device, which a lot of your projects probably will, they are going to interact with each other. Okay, they have to, that's how it works, right? So you will have what we'll refer to as the sensing device come first. It's going to be upstream. And then downstream of that is going to be your reporting device. Okay, so the sensing device, its job is to detect a specific signal, which is going to be probably your input, and it's going to convert that signal into some sort of transcriptional output. All right, and I'll show you what I mean by that in a second. The reporting device is pretty straightforward. It's just going to convert a signal that it gets into your output. All right, so let's define these for a second. So in our example, the signal device is our arsenic detecting device. And our reporting device is our color generator. Okay. When do we want the color generator to be on? Under what circumstances? Do we want it to be on all the time, producing color all the time? Or do we only want it to be on certain times? Yeah, good. I see arsenic in the chat. Yeah, so we don't want this to be on all the time. Oh, the chat's blowing up. Good. We only want the color generator to be turned on if our input arsenic is present. So we need it to be turned off, actually, until arsenic is present. And that's going to be the job of this sensing device, our arsenic detecting device in this case. So our sensing device here is going to act as a repressor. And I've drawn this symbol here, okay? That symbol represents stop or inhibited. It basically means that whatever is downstream of it has been inhibited or repressed or stopped, okay? So if there's no inducer, no input, in this example, arsenic, if there's no arsenic, then our arsenic detecting device is going to stop our color generator from operating. All right? Okay. Now, if we do have arsenic present, we have our input, that's going to turn off this inhibition, this repressor. Now it's like a double negative, right? It's going to turn off a repressor. So now we can get our color generation. Okay, does that make sense? I know it's like getting a little tricky now. Yes, all right, I got some yeses, good, okay, okay. I know, once we start doing repressors, it gets a little complicated. Okay, so that's getting you through your device level. So when we say device level, this is what we mean. You should be able to draw out a system like this, or you can do both, right? Where you have your input, you know what your input is. Here's arsenic. You've chosen your chassis. You know what your output is, and it does not have to be color. I know everyone loves to use color as their output. It does not have to be color. And you have your device or devices, and you're able to show us how those devices interact with each other if you have multiple devices. Okay? Okay, so in our second webinar, we will get into parts. I'm going to preview that a little bit so that you can see why we're not tacking this on to the end of this webinar here. So for parts, we want you to start looking at what the genetic components are that you're going to need to make those devices actually work. Okay, so we're not talking generic like, oh, it's a color generating device. We actually want to find real genetic components that you can use to build your machinery for real, right? And we're going to walk you through the different symbols here down on the bottom that you will encounter and will use to show us what your genetic parts look like. So you'll have a promoter and you'll look for a specific promoter. Promoters can be triggered by different things. So in the arsenic one, for example, there are promoters out there that are triggered by arsenic. So you could use a promoter that gets triggered by arsenic. You might use a specific ribosome binding site. Not very likely. Open reading frame is going to be the very specific code for the protein that you want. Kind of like when you're flipping through, or I shouldn't say flipping through, if you're scrolling through like a food recipe blog, you know how you have to like scroll forever and ever until you actually get to the recipe? The actual recipe is the open reading frame. Okay, it's not the rest of the fluff. It's just the information you need to make your protein. Okay. Okay, so in the end, your goal is going to be something that looks like this diagram here. We walked you through today how to do the top part. And that seems pretty straightforward, but you will probably change your project a million times and go back and forth and keep kind of fine tuning it, picking a better chassis. So hopefully by the time we do our second webinar, everyone has nailed down this top part. And we can kind of walk you through then how to get these parts put together. And we'll help you out with where you can find the parts, how they interact with each other, how to kind of lay them out in genetic circuits. Okay. So for now, you're figuring out your topic, you're figuring out your system, your inputs, your outputs, your chassis, and what device or devices you might want to use. And it's also okay to have several options because a lot of times we do this similar project on a bigger crunched timeline with our summer research program students. And sometimes they end up last minute switching to a completely different project that they had on the back burner. So it's totally fine to have several options running. In the meantime, resources that are great for brainstorming this stuff, of course, your BioBuilderClub handbook. It's going to walk you through even more examples than what I did with you tonight. Your BioBuilderClub book, that's a fantastic resource as well. The first 2 chapters really kind of hone in on what we talked about today. And then there are several real life examples in there as well. iGEM, of course, it has a plethora of SynBio projects designed by high schoolers. So you can check those out. And SynBio Beta is also another amazing resource for just getting ideas of project topics. And you can look at different devices and how they might work. All right, I think we've got about 10 minutes now for questions. I'll do my best. Chloe is the BioBuilder expert here. BioBuilder
I
Club.
just wanted to interject real quick. The item Jamboree just wrapped up, so hopefully soon we'll be able to hear from our BioBuilder Club students who also participate in iGEM. So congrats to all those teams for their amazing work, and soon you'll get to hear about their projects that they all just presented in Paris at the Jamboree.
I think I
And
saw.
also since, oh, yeah, is there a
No, I
question?
was just gonna say, I think I saw the Boss Lab iGEM team that we work with some research program got, I think it was best high school presentation. I think they got an award for that. They did a good job.
For sure, yeah. And since in the BioBuilder Club, all the teams you're working with mentors, right? So definitely once you have kind of a lot of the details of your system level design worked out, it's a great time that if you want to fill out the mentor request form, I can assign you to a mentor with experience in that subject area and perhaps with the type of chassis that you select. And so Danielle was saying, you know, there's so many different ways to end up building a system. And like in the summer research program, if teams, you know, are switching last minute, there's so many ways that if you're looking to eventually to build and test your designs, working with a mentor at that point is a great way to kind of select the approach that's gonna be the most kind of able to be built later on. So that's also something to keep in mind. Any questions?
Hi, Chloe. Hi, Danielle. Thank you so much for the presentation. It helped a lot. I started reading the book. This is my first time sponsoring this club. I'm the sponsor, but I have no clue. But I read the book, but your presentation helped me a lot to understand better. Thank you so much for that. I have a question. Like, for example, if you're thinking about the systems, like finally came up with your example, I'm going to take your example where you're taking arsenic detector. So where can we find them out? Like, for example, I have something like a heavy metal in my mind that I want to research about. So where can I go and look up like how to design them?
Yes, so oh wait, I'll just say next week I'm giving a presentation on literature research. So that will be all about how to find this kind of information, but I will hand it off to Danielle here related to the biodesign aspect.
I'm not sure what level of research you're looking for.
Like in general, if you have another heavy metal that you want to um, detect, like you said, you have to build something arsenic detector. So
Mhm.
something similar to that, what is the bacteria that we have to engineer that has a similar, so where can we find all that information?
Yeah, so in the BioBuilder Club handbook, the purple one here, that Chloe has written, there are a bunch of links. I think it's page, is it 17, Chloe? Did you say page
Yeah.
17? Yeah, there's links to a bunch of resources. And there's a website that actually is a huge database of parts, which we will get into in the parts webinar. But that's
Okay.
where you would find like, oh, I'm looking for a promoter that detects lead, right? That's where you might find
Yeah,
that. Or I'm looking
yeah.
for a chassis that lives in soil around pH 8, right? That's where you might be able to find stuff like that.
All right. All right. Thank you so much.
We have a question, Chloe. Okay, there we go. Okay, yep.
Yeah, I'll email out the link again. I did update the handbook last week, and so the link that I send out when you download it, anytime you download it, it'll be the most recent edition. But in next week's literature research presentation, I will talk about kind of like what you were saying about specifically SynBio related resources. If you're interested in something, how do you find more information on it, and also how do you find that information for free? Um, so we'll definitely go over that. And you mentioned heavy metals, and I will say that I can talk about this a little bit next week and things. But I have had some conversations with mentors about kind of some creative ways when working with designs that you couldn't necessarily just, test regularly in a high school lab. Probably can't just, bring in arsenic. So we can talk about some creative ways to research that because that actually did come up last week in our mentor meeting.
And
But if
honestly,
there's any oh yeah, sorry, go ahead.
I'm just gonna say, you can honestly just like Google stuff too, like bacteria that can detect lead or whatever it is you want it to do. And articles should pop up. Even if they're not free to you should still be able to access like the abstract or a little blurb about it to kind of like give you some ideas of what's out there that you can actually use.
Yeah, I think I'm taking one step at a time. Mostly I'll wrap my brain around by then.
Yeah, and if you have any specific questions or anything, feel free to email me. I'm happy to, provide more resources for specific
Sure, sure.
situations.
Thank you, Chloe. Thank you, Daniel.
And you can also look at what's already been done and kind of build off of that. You don't have to come up with some completely novel thing. Obviously, there are countless papers out there on trying to tackle the plastic pollution problem, right? We haven't figured it out, and there are so many different things out there. So you don't have to come up with some completely novel approach. It can be very similar to what someone else has done. You're just going to add in this piece, or maybe there were two different systems tackling the same problem, and you're going to see if you can kind of combine those two things and see if that works. I don't know.
That's an excellent point. A lot of teams email me saying, you know, oh, we had this project idea, but then we did some research and we found out someone did something very similar. And it's like, that's totally fine. Like, you know, like Danielle said, these problems have not been, you know, solved once for the last time, you know. So there's lots of different approaches, and even just modifying something that was done in the past, or doing like in a different cell type is definitely a completely valid project approach.
Or you can take a system that already exists and see if you can tweak it to detect something different, right? Or maybe the output is something different, but it's like the basic system is still intact that somebody else made. It's just, it's gonna sense something different now. So you're gonna have to change a few pieces of the parts, but you're not starting from scratch, right? It's kind of like the interior designer and architectural engineer, right? They don't start with just an empty plot of land. You usually hire them when there's a building and it's not laid out the way that works for you, right? And they're just knocking down walls and building walls, right? They're taking something that already exists and putting pieces in different places. And that's what you're doing.
And if you do some Googling and you find that it's specifically something that an iGEM team has worked on, and I'll show everyone next week how to do research on the iGEM teams, but they all have wiki pages, and you can definitely go see exactly how they've kind of built their designs and what parts they used. And so it can also be really good inspiration, even if your project is a little bit different, because all the wikis are really beautiful, and you can kind of see some more concrete examples that might be easier to read of how things were built than if you necessarily just pull up research papers. I saw some students just joining us now, so I hope that the time change didn't confuse people, because in most of the US over the weekend, the time changed. So we're in a different time zone now. Well, if anyone has any follow-up questions for Danielle, feel free to email me or Danielle at biobuilder.org. And this will get emailed out to all of the teams. We'll put the recording on YouTube so you can check it out. And yeah, so next week we will talk about literature research, and then later in the season, Danielle will be back to teach us about parts. All right, let's see. Thanks, Anna. And in December, Anna is going to teach us about plasma design. So that should be exciting. Okay. Well, if no one else has any questions. Anna, did you have any questions?
No, I do not. But that was really great, Danielle. Thank you.
Yeah, no problem.
All right. Well, thank you, Danielle. That was very helpful. Really appreciate that. And oh, yeah. So a couple people have asked to just send me an email real quick if people have specific questions, and I will talk to you guys soon. All right. Thank you, Danielle. Thanks, everyone.
Good luck, everyone.
Bye.